In this video, I'm going to discuss cell signaling. Cell signaling is really important because the cells, especially in multicellular organisms, need to communicate so that they can ensure that everything's in working order and the functions that are going on are what needs to happen. So what are the parts of the system? Well, there's got to be a cell that we're trying to signal, and there has to be a signal, and the signal is also often called a ligand. This is a chemical, but the chemical wouldn't do much if there weren't a receptor somewhere on or in the cell for it to bind with. One of the ways that we classify cell signaling is by sort of how far apart the signaler and the receiver are. If it's the same cell that is sending the signal and receiving it, we call that autocrine signaling. If the cells are near each other, we call that paracrine signaling. And if the cells are very far apart and the bloodstream is used to carry the signal from point A to point B because diffusion's really slow, we call that endocrine signaling. In addition to thinking about how far the cells are away from each other when we think about their signal, we have to think about where the receptor is located. And I want you to remember that if we think about a cell membrane, that the outsides of the membrane are hydrophilic, that means that they mix with water, and the inside of the membrane is hydrophobic, and that layer is actually much bigger than the outside. So when we have receptors, they can come in two different varieties. They can be what we call intermembrane receptors, where they're located within, or transmembrane receptors, where they're located within the membrane, or they can be all the way inside of the cell. And the type of signal that these two will receive is a little bit different. You can imagine if you have a hydrophobic signal, it's going to be able to get through the cell membrane because hydrophobic things can make it through that sort of big double stuffed Oreo of hydrophobic membrane. Those can bind to the intracellular receptors. Now, if we've got a hydrophilic substance, it's not going to be able to make it through that hydrophobic membrane layer. And in those cases, it's going to bind to the transmembrane receptor that's located extracellularly. This means it doesn't even have to go through the membrane to do what it's going to do. And you may wonder why this matters. It's not just something that instructors force young biology students to memorize. It actually matters because this kind of poises these receptors to do different things. Those transmembrane receptors can do something simple like open an ion channel and allow a new ion into the cell that wasn't able to get in previously. The intracellular receptors, those guys are actually able to interact with stuff inside the cell, things like DNA, and they're much more capable of doing things like turning on transcription and translation. The transmembrane ones can do this, but it takes a lot more steps for them to get there. But to understand your receptors, you really need to understand sort of the anatomy that underlies them. Receptors have something called a ligand binding domain. This is the part of the receptor that binds the ligand or the chemical, the signal. And this is specific. It's only going to bind certain compounds. Now, attached to that ligand binding domain, I think of the ligand binding domain like a light switch, and the thing attached to it is like what's plugged into that socket. This is called the functional domain, and the functional domain determines what that receptor does. So you can think about it like if you have a light switch, it could be attached to an outlet that's attached to a lamp, or it could be attached to an outlet that's attached to a blender. As long as that light switch turns on, it's going to turn whatever is attached to it on. That's kind of how these functional domains work. And the cool thing about this is that since these are proteins, they can be modified and they can have new functions and they can bind new ligands. Now, when we look at these transmembrane receptors, they kind of come in three different flavors. One of these flavors is ion channel linked receptors, and these, just as the name suggests, when that ligand binds the receptor, it opens an ion channel. This can cause all sorts of things to happen, we say downstream in the cell, as a result of these influxes of ions. But the receptor itself is just allowing them in. We can also have receptors which are called G-protein linked receptors or G-protein coupled receptors. In these, when the ligand binds, it's going to kick off sort of a chain reaction. Attached to this receptor are 3 subunits, alpha, beta, and gamma. Beta and gamma kind of go together, so I drew them as a single subunit. What's going to happen is GDP, which is very similar to ATP, is going to get converted into GTP when that ligand binds. This is going to cause beta gamma to go off, and that beta gamma can bind to something and cause signaling downstream. Alternatively, when that GTP is hydrolyzed, going back to GDP, this can cause alpha to come off, and alpha can go and signal. And these things can both happen when that ligand binds. And what this means is that these receptors can have a lot of different effects because there's a cascade of events happening downstream. This is called a second messenger because basically the ligand's the first message, the second message is whatever is happening downstream. And you might guess that these things take a lot longer for the cell to carry out, but they can be really potent because the cascades can be very large. A third type that exists is another kind of second messenger. It's an enzyme-linked receptor. What happens in these cases is the ligand binds. This causes the enzyme, which is part of the receptor, to dissociate, and now it can go and carry out its enzymatic function. This can cause all kinds of things downstream in the cell, and you can imagine what would happen if your enzyme was like an mRNA destroyer. It would be able to really change that gene transcription that's happening downstream. So I wanted to give you an example of one of these kinds of cascades that can happen. This particular one is a growth factor, and the type of receptor is called a tyrosine kinase. You might guess from the ACE, it's an enzyme-linked receptor. So basically, once the ligand binds, now that enzyme's able to do something. In this case, kinases are really good at adding phosphates to things. Here we have a protein kinase, which is called RAF, and that is activated by RAS. And I know these are always hard to remember, but just remember it's a kinase, so it's really good at adding phosphates to things. In this case, it adds a phosphate to this MAP kinase. Now, what that ends up doing is it actually is also a kinase. It adds a phosphate to this ERK. And the ERK is then able to enter the nucleus and do something. Notice that there are multiple steps here, and anytime you see multiple steps, one of the reasons that this occurs in biological systems is because it can be a source of amplification. So that MEK can probably phosphorylate multiple ERKs and have multiple ERKs go in. Another reason to have a multi-step cascade like this is that it can be blocked at any given point. So there's a higher degree of control in these cascades. So the kinases add phosphates. We call that phosphorylation. And phosphorylation has the ability to turn things on or turn it off. And it's because these sorts of chemical reactions can change the shape of a protein. Now, kinases are phosphorylating. They're adding phosphates. There's also dephosphorylation, removing phosphates. This occurs through a different kind of enzyme called a phosphatase. And these can basically turn on or turn off things that are going on downstream. This is a really cute drawing from Khan Academy, and basically it shows a similar cascade to what I've been talking about, which is protein kinase A. What's happening in protein kinase A is it's a kinase. It's going to add a phosphate to something. This allows it to activate that thing in this case and now create a cell response. Just as easily, it could be that adding that phosphate could turn the cell response off. So it's as often a way to turn things on as it is to turn them off. And so using these phosphatases or kinases, we can now either turn on cell functions or terminate cell functions as seen fit, just through the use of cell signals. One of the examples of a cell signal that can do something like this is calcium. If you ever fall asleep during a biology lecture and you wake up in the middle and you need to ask a question, ask what the role of calcium is. It's always a good question. The role of calcium can vary wildly, but it has some proteins that it interacts with. One of these is calmodulin, and this can cause downstream effects by phosphorylating or dephosphorylating things, and now it can change everything down to gene transcription, just depending on what exactly that receptor acts on. So here we're looking at another one of these cascades. In this case, this is a phospholipase C cascade. And basically what's going on here is, again, the ligand is binding, it's activating this lipase, right? It's an ACE, it's an enzyme, and this is creating a sequence of events. What I want you to notice about this particular sequence is, in this case, it requires both that IP3 to result in release of calcium, and the calcium to bind protein, which binds proteins, and the activation of the protein kinase C, which phosphorylates targets, to get the cell response. So it requires multiple things to happen for that response to happen. This, again, can be done for control. These cell cascades are super complicated, and there are many, many, many. The big thing, honestly, that I pay attention to is it a kinase or is it a phosphatase? And rather than focusing on what the acronym for each step is, it's more important to think about, what is that step doing? Oh, this enzyme is active, so it can change this thing. And not IP3 moves into its active state, which you might not remember what that is.